Welcome to the SWAYAM NPTEL course on Food Microbiology for Safe and Sustainable Food Systems. We were discussing beneficial microorganisms in food. Now, in this lecture, we will be discussing food fermentation. And the topics we will be covering: the introduction, fermentation, microorganisms involved in food fermentations like bacteria, yeast, molds, and types of fermentation, fermented foods, then history of fermented food, then advantages of fermentation, then lastly food fermentation, science and technology. Introduction. Food microbiology can be divided into three focus areas: beneficial microorganisms, spoilage microorganisms, and disease causing microorganisms. Beneficial microorganisms are those used in food fermentation to produce products such as cheese, fermented meat, that is pepperoni, fermented vegetables, that is pickles, fermented dairy products like yogurt, and ethnic fermented products such as sauerkrauts, idli, and kimchi. In fermented products produced by natural or controlled fermentation, microorganisms metabolize complex substrates to produce enzymes, flavor compounds, acids, and antimicrobial agents to improve product shelf life and to prevent growth of pathogens and to provide product attributes. Microorganisms with their enzymes also break down indigestible compounds to make the product more palatable and easier to digest. In addition, the beneficial microorganisms also serve as probiotics to impart direct health benefits by modulating the immune system to provide protection against chronic metabolic diseases, bacterial infection, atherosclerosis, and allergic responses. Examples of beneficial microorganisms are Lactobacillus acidophilus, Lactobacillus arabinosus, Lactobacillus lactis, and Pediococcus cerevisiae. Food spoilage microorganisms are those which upon growth in a food produce undesirable flavor or odor, texture and appearance, and make food unsuitable for human consumption. The microenvironment created in a spoiled food generally discourages the growth of the pathogenic microorganisms which are considered poor competitors. Now, the fermentation. Fermentation is one of the oldest biotechnologies for the production of food products with desirable properties such as extended shelf life and good organoleptic properties. Finished fermented foods usually have an improved microbial stability and safety, and some can be stored even at ambient temperatures. Furthermore, there are several examples of fermentation processes which lead to an increase in nutritional value or digestibility of food raw materials. Finally, food fermentation processes also deliver products with increased palatability for consumers. All these arguments have boosted the interest to explore natural food fermentation processes and more precisely to link the diversity of the community of fermenting microbes and their properties to the energetics of the process and to product quality. What is fermentation that is presented in this picture? The fermentation is a biochemical process that gets energy from carbohydrates and does not require oxygen. Here the products like ethanol is produced, the gas is also produced, especially the hydrogen gas which imparts fats, the lactic acid for example in cheeses, and the carbon dioxide for example in breads. And in this process the glucose is converted to ethanol, carbon dioxide and energy. From a biochemical point of view, fermentation is a metabolic process of deriving energy from organic compounds without the involvement of an exogenic oxidizing agent. Fermentation plays different roles in food processing. Major roles attributed to fermentation are: the first, preservation of food through fermentation, preservation of food through formation of inhibitory metabolites such as organic acids, for example lactic acid, acetic acid, formic acid and propionic acid, ethanol, carbon dioxide, diacetyl, rutin, bacteriocins, etc., often in combination with diseases, in combination with decrease of water activity by drying or use of salt. Secondly, improving food safety through inhibition of pathogens or removal of toxic components. And the third, improving the nutritional value, and the last, the organoleptic quality of the food. The common groups of microorganisms involved in food fermentations are bacteria, yeast and molds. The most important bacteria in the fermentation of foods are the Lactobacilli, which have the ability to produce lactic acid from carbohydrates. Other important bacteria are the acetic acid producing Acetobacter, mainly from fermentation of fruits and vegetables, and Bacillus from fermentation of legume species, and Bacillus species from fermentation of legumes. The beneficial yeasts in terms of desirable food fermentation are from the Saccharomyces family, especially Saccharomyces cerevisiae. Yeasts play an important role in the food industry as they produce enzymes that result in desirable biochemical reactions such as the production of wine, beer and ethanol and leavening of bread. The lactic acid bacteria, in short LAB, are however the most commonly found microorganisms in fermented foods. Their crucial importance is associated with their physiological features such as substrate utilization, metabolic capabilities and probiotic properties. Their common occurrence in food coupled with their long historical use contributes to their acceptance as GRAS, that is the generally recognized as safe for human consumption. The various lactic acid bacteria have been isolated from different fermented foods. Their functions during or after fermentation have gradually been elucidated. A vast range of materials are produced by industrial fermentation, and these include the organic chemicals used as fuels, food additives, antibiotics, enzymes for use in food and other industries. Vinegar is an example of a food additives produced by an industrial fermentation. Organisms are produced on a large scale for extraction of protein, for example, single cell protein that can be used as a part of human diet. Corn is an example of a single cell protein. It is produced from the fungus Fusarium graminearum. Yeast cell produced for use in industries such as baking industry, which relies on mass production of large amounts of baker's yeast. Foods produced on large scale as a result of the additives, as a result of the activities of microorganisms, for example, cheese, yogurt and bread. Production of alcoholic beverages, for example, beer and wines. Cellular extracts used as food additives, for example, yeast extracts from yeast cells produced as a byproduct of the brewing industry. Lastly, the industrial fermentations are now often considered under the heading of biotechnology, that is, the technology that uses living organisms and their products in manufacturing and service industries. The microorganisms involved in food fermentations. The most common groups of microorganisms that involved in food fermentations are bacteria, yeast and molds. The bacteria. Several bacteria are present in foods, the majority of which are concerned with food spoilage, while some like Clostridium are the causative agent for production of toxin like botulin, causing botulism in man. As a result, the important role of bacteria in the food fermentations is often overlooked. Lactic acid bacteria like Lactobacillus, Pediococcus, Streptococcus, Oinococcus, etc. are the most important bacteria in fermented foods, followed by Acetobacter species, which oxidizes alcohol to acetic acid. The acetic acid fermentation has been used extensively to produce fruit vinegars, including cider vinegar. A third group of bacteria of significance in fermentation are the Bacillus species, for example, Bacillus subtilis, Bacillus licheniformis and Bacillus pumilus, which bring about alkaline fermentation. Bacillus subtilis is the dominant species, causing the hydrolysis of protein to amino acids and peptides and releasing ammonia, which increases the alkalinity and makes the substrate unsuitable for the growth of spoilage organisms. Alkaline fermentations are more common with protein rich foods such as soybeans and other legumes, although there are few examples utilizing plant seeds. For example, watermelon seeds and sesame seeds are the substrates for alkaline fermentation. This picture depicts the lactic acid bacteria, what it causes. If it is consumed as a whole, then it serves as a probiotic. When it is disintegrated, it produces the intracellular metabolites like lipoteichoic acid that is released in the medium, and when it grows extracellularly, then the metabolites like lactic acid is produced and some bacteriocins are produced. And this picture depicts what are the different factors which affect the bacterial fermentation. For example, the performance matrix, the bacteria, if it is allowed to grow inside a matrix, then what will be the source of carbon, that will affect the growth. And after that, the fermentation medium, where it is growing, and whether it is in the sea water, alkaline medium or some other medium, that and what is the starter culture, that will affect the growth. Then the fermentation mode, whether it is the batch model or fed batch process or the continuous process, and also in what condition it is being growing, like for example, aerobic or anaerobic fermentation is going on. Likewise, the substrate feeding, whether it is a batch type or continuous type of feeding, and when followed by the fermentation scale up, whether it can be scaled up or it will remain as a batch process, and then the downstream processing means collecting the process of collecting the end products, etc. Now, the second most important example is yeasts. The yeasts and yeast like fungi are widely distributed in nature. They are present in orchards or vineyards, in air and soil, and in the intestinal tract of animals. Like bacteria and molds, yeast can beneficial and non-beneficial effects in food fermentation. Some of the yeasts like Pichia are viewed as spoilers of food products, while those like Candida are utilized for the single cell protein production. The most beneficial yeast in terms of desirable food fermentations are from the Saccharomyces family, especially Saccharomyces cerevisiae, involved in bread making and alcohol in wine fermentation. The Saccharomyces cerevisiae variety Ellipsoideus is employed extensively in wine making. Schizosaccharomyces pombe and Schizosaccharomyces boulderi are the dominant yeasts in the production of traditional fermented beverages, especially those derived from maize and millets. Saccharomyces cerevisiae variety Culbergensis is the yeast involved in beer production. Schizosaccharomyces pombe has been found to have capacity to degrade malic acid into ethanol and carbon dioxide, and has been used successfully to lower the acidity in the grape and plum musts. A number of yeasts like Rhodotorula, Cryptococcus have capacity to produce pigment to be used as biocolor. This picture depicts how the yeast cell that converts sugars into ethanol. At the same time, they release some energy, and this is the different pathway by which the sucrose is first degraded into fructose and glucose, and then ultimately they will go to the degraded to the glycerol 3 phosphate, and then it is finally it is converted to ethanol. The third most important microorganism is molds. Molds are also important organisms in food processing, both as spoilers and preservers of foods. Many molds have capacity to produce enzymes of commercial importance, such as pectinase by Aspergillus niger, Species of Aspergillus are involved in the production of citric acid from waste like apple pomace. The Aspergillus species are often responsible for undesirable changes in foods causing spoilage. On the other hand, Penicillium species are associated with the ripening and flavor development in cheeses, while the species of Ceratocystis are involved in fruit flavor production. At the same time, Penicillium is a causal agent for production of toxin like botulin. This is the again pictorial representation. This is the pictorial representation how the fermented foods are made. Like sugars with the lactic acid bacteria, they are converted to lactic acid, and the example is kimchi. Likewise, the sugars with the help of yeast, they are converted to alcohols and other alcoholic beverages. The molds, they are also grown to produce different types of microorganisms. And in this picture, the yeast and the bacteria in wild fermentation work together to prevent other microorganisms from growing. The yeast, they help breakdown of starch into easy to use sugars. These sugars, yeast, they will convert the higher carbohydrates to sugars, and these sugars can be utilized by the bacteria, and these bacteria will produce lactic acid and acetic acid, and this will hinder and lower the pH, and by that way protects from the outside microbes. And yeast also, which produces ethanol, and that ethanol is also have some antibacterial effect. Now, the types of fermentation. The various types of fermentations have been studied based on kind of respiration and mode of fermentation processes. Like aerobic fermentation. Aerobic fermentation is set of chemical reactions that involved in production of energy by completely oxidizing the food. The aerobic fermentation occurs in both cytoplasm and mitochondria. It occurs in higher animals and plants. The aerobic fermentation uses molecular oxygen as the final electron acceptor in the electron transport chain. It produces 6 water molecules per glucose molecule. Glucose is completely broken down into carbon dioxide and oxygen. The NAD regeneration occurs in the electron transport chain. The ATP is produced during the NAD regeneration, and during the aerobic fermentation 6 ATP is produced. ATP is the form of energy which is produced during the aerobic fermentation or the respiration. The another important fermentation is anaerobic fermentation, which is a chemical process in which the organic substrates are broken down to ethanol, lactic acid by the help of microorganisms in complete absence of oxygen. It occurs in cytoplasm, and the causative organisms are yeast, parasites and bacteria. It does not require oxygen. It does not produce water. The glucose is incompletely oxidized either to ethanol or to lactic acid. NAD regeneration occurs during the partial oxidation of pyruvate. The ATP is not produced during the NAD regeneration, and that is why only 2 ATP is produced during the anaerobic fermentation. Now, the batch fermentation versus continuous fermentation. The batch fermentation is a process where all the substrate and nutrients are added to at 0 time or soon after inoculation takes place, and the vessel is allowed under a controlled environment to proceed until maximum end product concentration is achieved. It occurs in batches. Conditions do not remain constant throughout the batch fermentation. Turnover is very low. Microorganisms use nutrients slowly. It is a closed system and log phase is shorter, whereas in the continuous fermentation the ingredients are added throughout the process as per the need and the products are removed as they are formed. It occurs at periodic intervals. Conditions remain constant throughout the continuous fermentation. Turnover rate is very high. Microorganisms use nutrients rapidly. It is an open system and the log phase is longer. In the lactic fermentation, the another type of fermentation, the lactose is utilized by the lactobacillus and it gains the energy, and then it produces different products like vinegar, kefir, that is the dairy products, then probiotics. These are the products which are produced. Or this is the alcoholic fermentation, in which the glucose is the main substrate, which is utilized by the yeast, that is the Saccharomyces cerevisiae, and it is converted to bread, cakes, biofuels. These are the main products. Now, the fermented foods. Fermented foods are those foods produced by the modification of a raw material of either animal or vegetable origin by the activities of microorganisms. Bacteria, yeast and molds can be used to produce a diverse range of products that differ in flavor, texture and stability from the original raw material. The production of many fermented foods involves organisms that are biochemically fermentative. Lactic acid bacteria that ferment carbohydrates to produce lactic acid are particularly important, but yeasts also play a major role in some food fermentations, fermenting carbohydrates to produce ethanol and other organic chemicals. Molds that do not ferment carbohydrates also play an essential role in some food fermentations. For example, the production of cheeses like blue cheeses and oriental foods like soya sauce. Now, the history of fermented foods. Fermentation as a food processing technique can be traced back to thousands of years. The history of fermented foods is lost in the antiquity. It seems that the art of fermentation originated in the Indian subcontinent in the settlement that predate the great Indus Valley civilization. The art of cheese making was developed as far back as 8,000 years ago in the fertile crescent between Tigris and Euphrates rivers in Iraq, at a time when plants and animals were just being domesticated. Later, alcoholic fermentations involved in wine making and brewing are thought to have been developed during the period 4000 to 2000 before Christ era by the Egyptians and Sumerians. However, the scientific rationale behind fermentation started with the identification of microorganisms in 1665 by von Leeuwenhoek and Hooks. Fermentation from the Latin word fever was defined by Louis Pasteur as a la vie sans la air, that is, life without air. Coincidentally, this was the time of the Industrial Revolution in Europe, which resulted in large scale migration of populations from villages to larger cities. There was therefore a dramatic shift from the food production for local communities to large scale food production necessary to meet the requirements of expanding and more distant markets. This in turn led to the development of large scale fermentation processes for commercial production of fermented foods and alcoholic beverages, with the most widely used microorganisms including yeast for the production of beer, wine and spirits, and lactic acid bacteria for variety of dairy, vegetable and meat fermentations. Modern large scale production of fermented foods and beverages is dependent almost entirely on the use of defined strain starters, which have replaced the undefined strain mixture traditionally used for the manufacture of these products. This switch over to defined strains has meant that both culture performance and product quality and consistency have been dramatically improved, while it has also meant that a smaller number of strains are intensively used and relied upon by the food and beverage industries. This intensive use of specific starters has, however, some drawbacks and can lead to production problems resulting in unsatisfactory strain performance. In case of lactococcal fermentations, bacteriophagous proliferations can affect cheese starter performance. This is the brief history of major events in the food fermentation. Like 6000 BC, the cheese production in Iraq following the domestication of animals started. In 4000 BC, the Egyptians discovered how to use yeast to make leavened bread and wines. In 1715 BC, Sumerians fermented barley and beer. Sumerian fermented barley to beer. In 500 BC, China, the moldy soybean curds as antibiotic they have started using. In 1776 AD, first whiskey distillery established in Ireland. In 1500s AD, fermentation of sauerkrauts and yogurt was started. In 1861 AD, Louis Pasteur develops the method of pasteurization. In 1928 AD, discovery was Nisin, which is antagonism of some lactococci to other lactic acid bacteria, shown by Rogers and Whittaker. In 1947, Nisin, the group N inhibitory substance, concentrated and named by Mattick and Hirsch. In 1953, the Nisin marketed in England and since approved for use in over 48 countries. In 1968 AD, FAO and World Health Organization recognized Nisin as safe and legal for use as a biological food preservative. In 1988 AD, the Food and Drug Administration of US approved the Nissin for food preservation. Now, the advantages of food fermentation. Food fermentations have been practiced for millennia, resulting the existence of a tremendous variety of fermented foods ranging from those derived from cereals, fish and meat to those derived from milk and dairy products. In each case, the fermentation process involves the oxidation of carbohydrates to generate a range of products which are principally organic acids, alcohols and carbon dioxides. Such products have a preservative effect through limiting the growth of spoilage and are pathogenic flora in the food products. In addition, a number of desirable products which affect the quality of the food may be produced, including the flavor compounds diacetyl and acetaldehyde, as well as compounds which may have positive health implications such as vitamins, antioxidants and bioactive peptides. When considering food fermentation as distinct from alcoholic fermentation involving yeast, the lactic acid bacteria is a primary responsible for many of the microbial transformations found in the more common fermented food products. Fermented foods are an extremely valuable addition to the human diet for a whole variety of reasons. For example, increase in variety. Fermented foods increase the variety of foods that are available, adding to our diet a group of highly nutritious products with unique characteristics. There are, for example, about 1,000 different types of cheeses. Secondly, the use of ingredients. Fermented foods form an important ingredient for a wide variety of dishes and are often used to impart special flavors. For example, pepperoni in pizzas, yogurt in curries, cheeses in a whole range of dishes including soups and soy sauce in stir fry dishes. Improvement in nutritional quality. The fermentation process may improve the nutritional quality of raw material. These improvements in the nutritional value of raw material will have little effect in the balanced diets of Western populations. However, for populations that subsist on diets consisting largely of polished rice, maize or other starches such as in Africa and Asia, the contribution that fermented foods make to the intake of B group vitamins and proteins is highly significant. Preservation. The fermentation often preserves a raw material, improving safety with regard to food-borne pathogens and increasing shelf life compared the shelf life of raw milk only a few days with the shelf life of yogurt several weeks. Health benefits. Some fermented foods are said to have definite health benefits, although the scientific evidence for this is limited. Reports suggest that fermented milk products such as yogurt can reduce serum cholesterol levels and help avoid cancers, particularly those associated with the colon. Bio yogurt, for example, AB and ABT yogurts are said to have a restorative effect on a normal microflora assisting recovery of a normal balanced flora after oral antibiotic therapy. Improved digestibility. Some fermented foods are more easily digested than the original raw materials. People who cannot digest lactose show lactose intolerance can often consume some types of fermented dairy products, particularly yogurts, without harmful effects. Detoxification of raw materials. The fermentation may remove toxic chemicals present in raw material. The cassava fermentation, for example, removes a carcinogenic, that is cyanide producing glucoside. Cassava is toxic if eaten raw. These are the different benefits of fermented foods, like it lowers the anti-nutrient content and boosts the nutritional value of foods. They keep skin healthy, they regulate the cell growth, they boost immunity and decrease inflammation, they protect against toxins, they help with weight management, they promote cardiometabolic health. Fermented foods support gut microbiome, they promote bowel regularity and digestion, they support a healthy mood, they help cognitive functions, they are appropriate for strong bones. The food fermentation science and technology. The science of fermentation is known as zymology. Food fermentation involves all those fermentation processes where either the ultimate product is used directly as a food, as an additive to food, or is a basic ingredient to the food or the byproduct formed during fermentation food waste utilization, their disposal or proper management. As a science, the food fermentation has an element of biological sciences, especially the microbiology, genetics and biochemistry. As a technology, the food technology, chemical engineering, along with integral component of sciences involved in food toxicity, acceptability and food nutrition. For many processes to make fermented food, essential components are raw materials, microorganisms, fermentation vessel and associated controls, processing, recovery and packaging systems. But from fermentation technology point of view, the fermenter and microorganism involved assumes prime importance. Since the end product would serve as a food, it is essential to evaluate it from nutritional toxicology and sensory quality point of view, and thus the knowledge of these aspects would form an integral part of food fermentation technology. The technological dimensions of food fermentation towards the application of engineering sciences in designing the fermenters and associated controls for optimum fermentation and product recovery are immense. As the product on the commercial scale would be marketed, which can occur only if it is economical, and therefore the economics of such products and the associated marketing aspects cannot be ignored. Similarly, transfer of knowledge to the scale of technology operation required is the paramount importance. The scale up operations, harvesting, biocatalysis of microorganisms, product recovery, effluent treatment, etc., are of concern to fermentation technologies. Food fermentation technology could be organized based either on the product or the system used to make the fermented foods or products derived from it, similar to food biotechnology. That is all for today. Thank you very much.